So hello, everyone. Thank you so much for joining us today. My name is Małgorzata or Gosia Kamocka, and I represent Indiana Center for Biological Microscopy. So today I would like to provide an overview of imaging modalities available at our center. And I will give lots of examples so you have a better orientation of what can be done using our microscopy systems. So we are a light microscopy facility specialized in fluorescence microscopy. So we provide investigators with access to various types of microscopy techniques, but mainly wide field fluorescence microscopy or laser scanning microscopy, which includes confocal microscopy, multiphoton, and also fluorescence lifetime imaging. We serve as a core for several NIH centers like the O'Brien Center for Kidney Imaging, the Diabetes Center, and the Center of Excellence in Hematology. So we support annually probably around 100 laboratories. It's very difficult to keep count of those, but we are pretty busy facility, having several systems and offering services like support with all aspects of microscopy. So we like helping investigators with designing the experiments, with conducting them. We provide training on microscopes, and we also help to analyze data and interpret them. So we actively developed methods of intravital microscopy of several organs, and this is done under the centers, NIH centers, which I mentioned. So basically, there are two imaging modalities which we offer in our facility. There is wide field fluorescence microscopy and laser scanning microscopy. Within laser scanning microscopy, we have single point scanning systems, confocal systems, multi-point scanning confocal microscope, which is called spinning disk confocal microscope. We also have multiphoton microscopes and fluorescence lifetime imaging systems. So we mainly, I have to say, focus on laser scanning microscopy, and you may ask why. Well, when you look at the image here, you can see that wide field microscopy doesn't give us really detailed information, doesn't have the resolution which we would need to see, to focus on the structure of our interest. And it happens because there is out of focus emission light contamination coming into the image. So we cannot really see all the details, whereas confocal microscopy gives us an image of one focal plane. Therefore, we can distinguish structures within that image. So this optical sectioning capability is really very important, and I would say that is the main advantage of confocal or two-photon microscopy. Those microscopes have better resolution, they have increased sensitivity, so they have good detectors. You can adjust speed, you can adjust, you can zoom into the area of interest, you can image multi-labeled specimen, and then don't worry that much about crosstalk between fluorophores because these microscopes have an option of eliminated crosstalk. So all of this makes laser scanning microscopy much better. And on top of that, we collect digital images which you can quantify, and also you can generate very nice 3D reconstructions. So the 3D optical sectioning, which is referred as a Z-stack, is a series of allows us to collect a series actually of focal planes and stack them together. So this is called a Z-stack. You can space them between, you can space them or you can collect them overlapping one another. So you have very nice Z resolution of the volume. But you can image it at one time point, but also you can image them at different time points, having nice time series of 3D volume. So both confocal and multiphoton microscope, they're able to do optical sectioning, but they work a little differently. So in confocal microscope, as I mentioned earlier, talking about wide field fluorescence microscopy, there is out of focus illumination which we have to deal with. So when I want to focus, so let's see that I illuminate the fluorophore in this cuvette with a blue laser, and I would like to image only one focal plane of this volume. So in wide field fluorescence microscopy, this out of focus light, this cone of emission generated above and below focal plane makes the image really very blurry and doesn't allow us to show to image details of interest. So confocal microscopes have a pinhole or confocal aperture positioned in front of detector. So all this out of focus light is eliminated. So I want you to understand that it's not excitation which makes the confocal microscope able of collecting one single focal plane, but it's a pinhole on the detector side which eliminate out of focus light. On the other hand, in two-photon microscopy, which is the image on the right, we are able to image only one single focal plane, but it happens on a little different basis. So when we illuminate the volume, the fluorophore with IR laser, the excitation will happen only in the small spot of sample. And it happens because only at that particular plane, particular spot, there is enough concentration of photons to cause excitation, and we need 2 photons of lower frequency, higher energy to lower energy to cause excitation. So both 1 photon and 2 photon excitation, they don't eliminate light scattering. So there is still an issue of imaging deep into the tissue. So what I want to talk about today are some applications. And I know that you probably joined previous seminars provided by Dr. Dan or Connor, so you may know our systems a little bit, but I feel like there wasn't enough information or examples about different type of applications. So generally, in our facility, we can image fixed specimen or live specimen. We do live imaging of cells. We also do live imaging of animals when we expose an organ of interest for imaging. And we also do ex vivo imaging. So let's say you can extract retina from the eye, put it on a dish, and image it for a while the cells are still happy and reactive. So what imaging we do are generally, they cover studies of histological structure. So it's very important to find out what is the composition of the tissue, what is the relation to other elements of the tissue. So having 3D information allows us to make judgments of the histological structure of the tissue. We also can evaluate physiological function, but for that, obviously, we need to have intravital imaging. What I wanted to focus today a little more is some imaging techniques which allow us to evaluate protein interaction and protein degradation. So, and finally, I will talk a little bit about biomarker signatures, setting imaging, which we can do with multiplex phenocycler system. So all of these applications can be done because we have several imaging modes which we can use to image samples. So first of all, we can image single optical section, and we refer to that type of acquisition as 2D imaging. But we can also do a Z series, so it means a Z stack, so a series of single plane images throughout the volume. We also can keep imaging it over time, so we can do time-lapse imaging. So besides single field imaging, we also can do multiple not adjacent fields imaging, and we can maybe register them in the microscope and come back to the same spots over time. And also quite popular technique is doing tile acquisitions. So those acquisitions are actually very useful because you can scan a big area of tissue with higher resolution, and those images consist of several adjacent fields. So then we have our confocal systems. Actually, one confocal system can obtain also transmitted light image of DIC. This is differential interference contrast technique, which I personally like very much. So I will give you example of that one as well. And it allows you to visualize cells or tissue without labeling. So it gives you 3D appearance of the sample. So then I will talk a little bit about fluorescence lifetime microscopy, and we have two flame microscopes, confocal and 2-photon. I will talk also about FRAP imaging. Actually, not that much of the FRAP, but I will touch base on that a little bit, talking about protein degradation. And then I will talk about fluorescence energy, resonance energy transfer, or Fromster's resonance energy transfer, to be more correct. So talking about confocal 3D imaging. So what is the best example of 3D imaging on a smaller scale, but looking at the spheroids and cysts? Many people, many investigators I see are growing spheroids in matrigel, and they like evaluating certain components of these spheroids under different treatment conditions. So the image on the left is the co-culture of pancreatic tumor cells and tumor-derived fibroblasts, which are in green. They are expressing GFP. And Dr. Fischel was interested in tracking these spheroids over time to see how they respond to different anti-cancer treatments. So you can really see very nicely how cells, cancer cells, which are marked in red color, disintegrate during successful treatment. So the next example would be a cyst. So those were not very big cysts, but when you look at the cyst in 3D, or maybe in wide field microscope, if you would like to, you would see just a blob, just a sphere there, and you wouldn't know what's happening inside. And when you look at the orthogonal view and display one single focal plane coming from the middle of this sphere, you can see that those are empty structures inside. And we can say that clearly because we can see the wall of this cyst coming on the other side. So these orthogonal projections, X, Z, and Y, Z, allow us to see all the structure of the spheroid from the top side and from the bottom of the spheroid. So those are the simplest example of 3D imaging, of necessity of 3D imaging, which confocal and 2-photon microscopes can perform. So I also wanted to show you an example of 3D imaging, which I've been doing with some investigators from the dental school. And they are interested in evaluating different treatment for infections, various types of infections. So over the years, I've worked with many different specimens with them. And here I wanted to show you very interesting, actually 3D image, which I collected of a sample, which it is a bovine enamel sample on which was mounted the orthodontic bracket. This is a low magnification imaging, but it gives us enough information to see where the bracket is and how the structure looks around. So on these brackets, on this specimen, they grown a bacterial biofilm, and they evaluated different type of treatment. And And you can image, it was impossible for us to image it with higher magnification. It would really takes lots of time, but this low magnification image gave us enough information to calculate the ratio of live to dead bacteria. The biofilm, they grow also biofilms on other specimens. I imaged them on metal discs, on implants. I also imaged the root canal. So all of this imaging is done of live bacteria. There are certain portions which are dead, of course, but we do live samples here. And these nice 3D reconstructions shows very big clusters of live bacteria and just a few of them which are dead, meaning that treatment in this particular experiment wasn't really very successful. And then, oops. And then I wanted to show you an example of large-scale imaging. So as I mentioned earlier, having only a single field, a representative single field, sometimes it's not really very helpful. So even if we collect several fields from the section of the tissue, it may not provide enough information. And also, we always select certain areas. So it's our judgment sometimes which interferes with the experiment. So many investigators collect tile acquisitions. So you can, this is very helpful technique because you can collect adjacent single fields and with certain overlap to be able to stitch them at the end. So this generates a nice image of the entire section, and you can do just one single focal plane, or you can do 3D volumes as well. But most importantly, we can always zoom in into this structure, like I did here, into this small area and show higher resolution image because each single field was collected with higher resolution lens. Actually, in this case, was only 20x objective, but still, it's a very nice image, and this is free color, multicolor image. So all of this can be done quite easily. And then DIC. So differential interference contrast is a technique which I like very much. So the beauty of this technique is that you don't need any labeling to visualize cells. It gives you very nice 3D appearance. But also you can you can overlay fluorescence confocal image with DIC. So you can collect them simultaneously. And I like this technique very much because it's so important to see a morphology of a cell. So sometimes it happens that, especially for live cells imaging, like we have here, that we label only certain structure of interest, but we don't want to label nuclei or other structures, sometimes even membranes, to don't influence the process, the cell, the metabolism of the cell. So in that case, having DIC image gives us an idea if cells were happy, if they were really creating a nice monolayer. Like, it's very nice to have this information. And I have to tell you that it happened to me many times that when I imaged some samples, live cell samples, that they were infected with mycoplasma, and investigators didn't know about it, but DIC image can show you clearly intrusions in the cytoplasm, so you can really make that judgment quite easily. So it's always helpful to have this information, and it also helps you to see if some fluorescent signals comes really from the cell or maybe from the area where cells were not present. So it is one of my favorite techniques. So we do live cells. Typically, we image live cells with a spinning disc confocal microscope, which is set up for live imaging. It's equipped with the incubation chamber. That way, you can image cells over extended period of time. It may be a few hours, it may be an overnight, or it may be just entire weekend. So this is just a very short video showing you movement of ER in a cell. Let me play it again because I know I made it super short. Oops, maybe I cannot. So it really, it collects images even at 30 frames a second. Of course, it would be one channel acquisition, but it's a very, very sweet system. And actually, I had to mention that one because it's my boss's favorite microscope. So I had to include that image as well. All right, so now I will switch to intravital microscopy of small rodents. So our facility is specialized in intravital microscopy of small rodents. We do really lots of intravital imaging. And in most cases, an animal is opened up in order to expose an organ or tissue of interest. But the problem is with this technique that we cause wound of the incision site, and we really cannot image it longitudinally because it's typically a terminal procedure. So on this picture here shows you a mouse prepared for bone marrow imaging. So preparations is typically quite simple. So we make an incision, and then we expose organ of interest. Here we image through calvarial bone. So just by doing simple incision, we are ready for acquisitions. If we image a kidney, we have to make an incision and externalize it just a little bit to be able to image a live organ. So the other possibility for intravital microscopy is to do longitudinal acquisition. So for that, we have to we have to prepare a specific model of animal. So there are techniques of imaging windows, which have been quite popular for longitudinal imaging studies. And there are some of them which we've actually used in our facilities. So there is a cranial imaging window. We tried it for the bone marrow imaging of calvarium, but it's also used for the brain. We've tried a dorsal skin folder chamber. I actually tried it myself for clot formation, but not very successfully. But another investigators who was interested in imaging the skin, wounding of the skin, had successful studies. And then we have abdominal imaging window and mammary imaging window. So in our facility, we do studies with abdominal imaging window. We place it over the liver, over the pancreas, and over the kidney. So you can anesthetize animal, position it on the stage, image the organ, and then come back to that same animal over a time. And here are some examples of intravital imaging. So on this slide, I included two videos showing clot formation in the mesentery venule. So the top picture shows the plasma flowing through the vessel, which is labeled in red, and the endothelium on the side of the vessel was injured with a laser. So clot starts forming, and there are platelets which accumulate, which are labeled in green, and they accumulate on the surface of the injury, in the site of the injury, and there is also fibrin labeled in blue, which gets accumulated. So not labeled blood cells are visible either as these black streaks, or they are visible as these shadows of cells. When they slow down enough, we can see them attaching, rolling on the endothelium. So we see nice leukocytes here. And here is another example of the stasis model. So in this case, the clot is formed very differently. We also labeled platelets. We should have fibrin accumulation here, but just a little bit. And you can see that those two these two structures are very different. So this intravital acquisition was used to create a computational model of thrombus development. So basically, when we acquire images, we do some kind of processing. We analyze certain, we analyze images for intensity or volume or distances. But in this case, we wanted to go a little farther, and we wanted to create a computational model of thrombus development, and it was a multi-scale model. So from our intravital imaging, we provided input into this model, so we could evaluate, we could put in various parameters into the model and predict what would happen with the growing thrombus under different circumstances. So this work was done by collaboration with mathematicians from the University of Notre Dame. And I have to tell you that at the beginning, it was really very challenging to communicate with them. It was almost as speaking a different language. But after a little bit, we really made a nice progress, and we were very proud of creating this computational model. Before that one, there were some models which people generated based on ex vivo imaging of platelets aggregation, but really they were not as sophisticated, let's say it that way, as our model. So always think about image analysis, not only in terms of looking at intensity or just, don't close your possibilities. Don't make it very narrow, because sometimes you can expand it to something really very different, very useful, and even unexpected. Okay, another example of intravital two-photon microscopy is a kidney imaging. So we did in our facility lots of kidney imaging, and Ruben Sandoval is our expert here for intravital kidney imaging. So he collects, he imaged probably thousands of kidneys by now. And I wanted to show you one of his images showing or time series showing filtration in glomeruli. So glomerulus, the capillaries of the glomerulus and other capillaries are visible by injecting high molecular weight dextran, which is not filtered, so it stays in the vasculature. And then Ruben injected low molecular weight dextran, which was labeled with blue fluorophore. So it gets filtered, let me play it again, it gets filtered from the glomerulus, it goes to the tubules, and then finally it accumulates in collecting duct, distal tubules and collecting duct. So you will see accumulation of blue signal over there. Ruben additionally injected mitochondrial probes into these animals. So we see mitochondria labeling distal tubule and collecting duct. This was done with TMRM. And then he also used rhodamin 123 to show other tubules which do not pick up TMRM. This image is just a 3D rendering of the glomerulus, showing again capillaries in a pink, nuclei, and some of the tubules here. And of course, from these studies, we can do all the quantitation of function. We can evaluate filtration. We can also evaluate tubular uptake. But on top of that, we can do regular structural analysis. So I am showing here at the bottom an example of structural analysis of the glomerulus. So this is the glomerulus, the capillaries, the vessels are visible in red color, and in blue there are podocytes. So I segmented all the capillaries, and then I could look specifically at the capillaries of the glomerulus, and then I could also segment podocytes. So this type of image analysis allows us to see, to evaluate volume, distance, intensity, and we can gather lots of statistical parameters out of this type of image analysis. So another example of intravital imaging is two-photon bone marrow imaging. You may know that it's not possible to image a bone marrow of long bones because our systems cannot image that deeply into the tissue. So the model of calvarium, imaging calvarium, has been quite popular for intravital imaging of the bone marrow. And we prepare animal in a simple way, as I already said, by incision and exposing the calvarial bone, which is very thin, so we can image through the bone and focus on the bone marrow. And this image was done, it's a mosaic image collected from a live animal, which was done by second harmonic generation of collagen. So it shows us nice outline of the bone. And then a big mosaic in the middle shows some fluorescence for the flowing blood, which is labeled, the plasma was labeled in red color. And we also see green cells in the niches, which are, the animal was, is a lice GFP animal, so myeloid lineage has GFP signal here. So we typically focus on 6 regions within the big area of the bone marrow. The main reason is that this area is the most flat, so it's easy accessible for imaging. And for each of the field, we can collect a Z-stack, we can do time series, so we can look at various parameters from this type of imaging. And a good example would be the type of analysis which Dr. Strauer done with intravital imaging. So he was specifically interested in calculated distances from cell to the surface of the bone. So we did 3D intravital imaging with live animals. We labeled vasculature by injecting high molecular weight dextran, and then we injected his cells of interest, and we're looking how they home into the bone marrow, and then we looked specifically at distances, volume and distances between the bone and the cell. Right. So so you could, we all probably agree that we cannot image 3D structures like cells, organs, or entire organism by sampling only a portion of the structure, by collecting 2D images. So we really would like to image the whole structures, but unfortunately, our microscopes are not capable of imaging through thick tissues. So there are several approaches allowing us to do this type of imaging, and mainly people do sectioning approaches, which can be done by doing serial sectioning of the big structure, like I presented in this picture, where I was specifically interested in looking at the clot formation in a carotid artery, which is big, and actually even the wall of the artery is too big to image through. So the only approach I could take was to section this artery after a clot was formed and image each of the sections separately. So I thought that having 30 micron sections would be a good idea, because this is the volume which I can quite easily image in confocal microscopy. But let me tell you that this technique really, it wasn't that great, because it generates lots of error. There is error with sectioning, so you really lose some tissue just by doing sectioning, and then they can be folded, they can be positioned not correctly, so that is very challenging. So even though I was able to collect all these volumes from the structure, I wasn't really very successful with putting it together manually, because it was even before time when we had the software allowing us to do this type of reconstruction automatically. So then another technique is block phase imaging, where you have a block of tissue, you image it, and then you shave it and image again. But the best approach for imaging would be probably non-sectioning approach, which is possible by using optical tissue clearing techniques, and that's what I would like to talk about. So you may see, okay, we image organs, we do intravital microscopy, so why do I need to do optical sectioning? I mean, optical clearing, sorry. So you may be right, but unfortunately, confocal imaging doesn't penetrate through depth very well, so we probably can image up to 30 microns. Two-photon microscopy, which can image a little deeper, probably for the kidney, which is presented here, can go up to 60 microns. But if we do optical clearing, we can go much deeper. So it is a kidney cleared with scale to solution where I was able to collect volume of about 600 microns. And I really didn't fully clear this sample yet. So the image on the right shows you the heart, which was cleared with a cubic solution. So Dr. Sumpa perfused entire heart with cubic solution and labeled it for nuclei, which are the main interest of his studies. So he was specifically interested in looking at cardiac myocytes. And you can see that we were able to image up to 2 millimeters through this volume. And free working distance of an objective set the limit for this imaging. So if we could image farther, but unfortunately, the objectives we have didn't allow us to do that. And there are many clearing techniques available, but if you are wondering what it is, it's actually a technique which makes alters the scattering properties of cellular components. So they create uniform density of scatter. So lateral scattering is minimal, and then we can have a wavelength of light passing through the tissue. I want you to understand that the optical clearing is not to eliminate scattering. The light scatters in heterogeneous environment, only in a vacuum it doesn't. So we just make it uniformed, I would say. And there are many sources of scattering: the membrane, lipids, our mitochondria, cytoskeletal, all these structures, cellular structure, or the tissue components scatter light a lot. So that's why we are not able to image deeply. So on this image, there is a brain, entire brain imaged before tissue clearing and after. So you can see that after tissue is cleared, we can read what the text underneath the tissue. And there are many tissue techniques available. There are solvent-based tissue clearing techniques. There are aqueous-based clearing techniques, and there is no one for all. So actually, you would need to do a little bit research to find out which one would be applied for your tissue and for the specific fluorophores which you would like to use, or type of labeling. And here is an example, my favorite example of tissue clearing, which was done by Dr. Ferkovich very long time ago. He cleared the entire mouse embryo, and he was looking specifically at progenitor cells labeled with CD41. And the beauty of this image is that he did it all manually. So it was done, I believe, at the time that we didn't have motorized stages. So he was imaging single fields, moving the stage manually. And we didn't have objectives with long working distance. So he placed this volume between two covered glasses, and he imaged from one side and then from the other, and he put it together. That's why I appreciate this image so much. And here is an example of cleared bone. So because we cannot image very deeply through the bone, and we really would like to see bone marrow in the intact in the whole tissue environment. So the clearing technique can be useful. I have to say that the bone marrow is very difficult to image. It has such strong endogenous autofluorescence. So on top of being positioned within the bone, which is difficult to image through, it also is difficult to image by itself. So this tissue clearing technique was cleared with cubic, and I was working with Roy from Dr. Strauer lab, who was really persistent in doing optical clearing, and he labeled the entire bone with topo-free or maybe cytox for nuclei. And then he also did some labeling for megakaryocytes and osteomax. So in that image, you can see, in that projection, you can see that there is an image of the bone, which we have based on second harmonic generation of collagen, which two-photon microscope allows us to do. And we also have multi-channel imaging. We see components of the cell, we see megakaryocytes, and we see in yellow some osteomax. So it is quite successful, but to be very honest, not as much as we would like to, because there is, due to high autofluorescence, we didn't achieve the results which we were hoping for. All right, so the next, if you have any questions regarding the tissue clearing techniques, I am happy to answer questions now, because I will switch to protein degradation methods.
Josie, there's a question in the chat.
Yes.
Which substances were used to clear the brain?
The first brain tissues were cleared with scale A2 solutions. So there was a Nature paper specifically for the brain, and evidently they were able to do very nice clearing while preserving fluorescent signal. But nowadays there is a boom of different clearing techniques. So I would say that look at the literature, what people use nowadays, because I know that cubic has been very popular, for example. I'm not sure if specifically for the brain, but I would suggest going to PubMed and search. I know that that a scale solution was originally used for the brain, for clearing the brain.
Another question, does your core help with the optical clearing techniques, or is it only imaging of cleared tissues?
Unfortunately, it's only imaging of cleared tissues. So we can help you. We can help you to choose the right method. We would have to probably do some research ourselves, but we can do that. And as in the process, you can we help you with imaging as well, because it's very important to choose correct objective, correct microscope to make sure that you can evaluate properly clear tissue. We do not have SPIM microscope yet. We may have next year, so we are still limited to this 2 millimeter working distance, so 2 millimeter volumes as of now. But next year it may change, and we may be able to image entire organs as well. All right, so let me switch to protein degradation. So I wanted, talking about protein degradation gives me an opportunity to mention optical highlighters. I really didn't see much interest in optical highlighters in our facility, and it's very surprising. So I wanted to show you one study which I've done a long time ago with Dr. Bascotto from Italy, who was always very happy to look at new modalities, new possibilities of imaging, and he was very enthusiastic. So we did wonderful studies with optical highlighter, which allowed him to determine half-life time of the protein of interest. And he also did PLA assay, which I will talk about in a moment. Commonly used methods for evaluating protein degradation or half-life time are not the best. There is radioactive method, which requires radioactive labeling, and there are also biochemical methods which are used with CHAs X treatment to inhibit protein synthesis. So because you block synthesis of all the proteins, cells typically don't do very well. They go for apoptosis, so the technique is not very great. And then there were fluorescence techniques like FRAP, so fluorescence recovery after photobleaching, while people were trying to bleach the area of interest and then see how the intensity recovers. The only problem is that to bleach the area, the region of interest first. You have to apply high power of the laser. So the cells are typically not very happy with that, but also there is a mobility of proteins within the cell. So it can be just, income of new protein to the region of interest, not necessarily indicating a synthesis of a new protein. So those techniques were not great, but optical highlighters come very handy here. And with Dr. Vascotto, I used Dendra-2 to determine lifetime of endonuclease. And there are different type of optical highlighters. They are photoactivatable, where they have no fluorescence at the beginning. You have to expose it to UV light, and then they become fluorescent. They are photoconvertible, so they have one color before photoconversion, and you can change the color after and track the cells over time. And then photoswitchable, which have emission characteristics that can be turned on and off. And then fluorescent timers, so the color changes as time progresses. So with Dr. Vascotto's, we used Dendra-2, as I said, and he actually expressed AP1 as a fusion protein with Dendra-2 in APE silent cell clones. So it was very important to do that because we wanted to maintain physiological expression level of the protein. So if you have, let's say, fusion protein with GFP, typically it is synthesized on top of non-fluorescent proteins. So you generate not physiological levels, concentration of this protein. So you should be careful with this type of imaging. So for Dendra-2, before photoconversion, it has green emission, so you can image it with 488 excitation, and after photoconversion with UV light, you can image it also with 488 laser, but the emission which you collect would be in the red spectrum. So the color changes from green to red as time progresses. So based on this imaging, Carlo was able to calculate AP1 half-lifetime. So I also wanted to mention that cells were doing well over a time. Even after 15 hours, they started producing synthesized new protein, which was again green. So it was a very nice study. And I would suggest looking into these optical highlighters because I don't think that they are used really enough. So then for protein interactions, I wanted to talk also about protein interactions. So many people coming to the facility, they want to do colocalization study, and they think that it gives them an idea of how it answers the question if the protein of interest interact or not. Unfortunately, it is not true because the resolution of the microscope is not high enough to determine if proteins interact with each other or just are present in the same space of, let's say, 0.2 micron. So we have to look at other techniques which would allow us to see interaction in the smaller proximity. And FRET is one of the techniques which is sensitive to a distance. So for FRET imaging, we have two probes. One is donor and one is acceptor, and both are connected to two proteins of interest. So only if they are in close proximity to each other, the FRET signal is generated. So this Jablonski diagram shows very nicely what happens during FRET. So in normal fluorescence, after electron jumps to higher energy state and it comes back to a zero grant, it emits fluorescence. When the donor, the appropriate donor, is present in close proximity to these, when acceptor is present in close proximity to the donor, the signal, the emission is transferred to the acceptor, causing excitation of acceptor, causing excitation state of the acceptor, and then we observe emission in a different energy. So here is another representation, one actually example of FRET to study proteolysis. So before two proteins which have fluorescent tags here are proteolyzed, they are FRET positive, they are in close proximity, so energy transfer can happen, and we observe emission of the acceptor. But after they are proteolyzed, we don't have FRET signal anymore because they are in not in close proximity. So this is a very powerful technique to look at protein interaction, but it may be quite difficult. And there are many techniques which you can do. And typically we suggest doing not only one, but maybe confirm one with the other. So if you want to do like 2-channel ratio imaging of donor and acceptor, and you excite only donor, not acceptor, but you collect emission for both, then I would probably suggest doing spectral imaging on top of that, or perhaps fluorescence lifetime imaging, which is also a very nice technique to look at FRET signal. And in this image, you have in grayscale just a regular confocal image looking at the ratio intensity of a donor and acceptor emission. But on the right side, you see fluorescence lifetime imaging. So very quickly, even before I can see any intensity changes in regular confocal image, I can see that lifetime changes. So FLIM, fluorescence lifetime imaging, depends on environment. And also FRET changes fluorescence lifetime. So it becomes, the donor's fluorescence lifetime becomes shorter. So this technique is actually very nice because we can image only donor and look at the changes of fluorescence lifetime of the donor. We don't have to even think about imaging additional acceptor or doing any type of the ratio. So then we have proximity ligation assay, which is another technique which allows us to look at the interaction of the proteins. And I really like this technique because it's very simple, and it allows us to look at true interaction between proteins, because PLA assay, the signal happens only if two proteins are in the proximity of 40 nanometers or below. So the technique is actually made based on special PLA probes. So you have to have primary antibody, which are specific to the protein of interest. Then you use special PLA probes which have oligonucleotide tag, and after this step, you add another oligonucleotides and you do hybridization. So this hybridization allows you to switch to a ligation step. So only if there is close proximity between two protein, this hybridization, this ligation step is happening, and you can amplify this oligonucleotide circle and then tag it with oligonucleotide, which are labeled with fluorophores to see positive PLA signal. So the results of this technique is the single spots, which you see in this picture. So you don't see the structure per se, but the single spot indicates that there was interaction of the protein in your sample. So Dr. Vascotto used this technique to look at the interaction of AP1 and NPM, nucleophosphine, and he could see interaction in the nucleus as he expected. Dr. Petruska quite recently was interested in looking at interaction of two proteins in myeloma cells. And so she did PLA assay. So these green spots indicate positive PLA signal. She was interested if signal was in the cell or not. Additional, on the signal, she wanted to calculate how much signal comes from each of the cell. She also was interested in looking at mitochondria and evaluate how many PLA assay is localized in mitochondria or outside mitochondria. So we segmented mitochondria. We also classified them based on sphericity, so we can specifically look for the signal coming from mitochondria or outside. So green signal here indicates that we had two proteins interacting in the mitochondria. And also we did the same for the nucleus, looking how many PLA signal comes from the nucleus and how much is coming from the cytoplasm. So fluorescence lifetime imaging is basically a technique which allows us to look not where the fluorophore is located and look at fluorescence intensity, but we are looking at what time the fluorescence is emitted. So there are two fluorescence lifetime measurements which can be done, two techniques. One is fluorescence lifetime time domain fluorescence lifetime microscopy, when you basically apply the pulse of laser and you look when the photon arrives after the pulse of laser. So there is a time domain technique, and then there is a frequency domain technique, which apply modulated source of light, and also emission is as well modulated. And instead of using the time gating to look at time arrival, we are looking at the phase shift and modulation of amplitude between excitation and emission. So this is another type of technique which we can do, but then we can do all the analysis. So we can create histogram of photon arrivals, and based on that we can do analysis like a curve fitting from which we can evaluate the lifetime. The other option is to do phasor plots or polar plot, which is basically a representation of grouping of vectorial representation of sinusoidal wave, where the photons, where the pixels having the same lifetime are grouped together. And there is, if there is one component of the lifetime, it will be present on this semicircle. If there is a combination, it will be within that semicircle. So lifetime microscopy is really very useful. First of all, for Fred, as I said before, and we can do in our system also pi Fred, so we can look at two biosensors, let's say, at the same time. But I found it also useful at distinguishing different components in the structure, which I am not able to distinguish using regular imaging. So here is an example of two-photon imaging, the green and red channel which we've collected. You can see that it looks pretty uniform, but we know that there is a bone marrow there having different type of cells, which we are not able to distinguish even after we apply certain staining because of high autofluorescence. But doing lifetime imaging, we can really group together pixels having the same lifetime and look how many components we have from red channel and green channel specifically. So there is a nice technique to look at more probes that we really can image. And the last technique, which I would like just to mention, because I am not expert from this technique, there's Connor in our facility who does multiplex codex imaging. He helps many, he has helped many investigators with this technique. And the beauty of this is that you can label the same section with many fluorophores. Traditionally, conventional technique gives us limitation to about 4 fluorophores. I would say you can try more, but it becomes more challenging. So in Codex, there are a special, they implemented special barcoding technology where antibodies have oligonucleotide barcode, and they work with, they are visualized with a reporter having fluorescent tag. So the phenocycler fusion systems, and we have 2 of them, allow you to set up a antibody panel and image by cycles of labeling, imaging, and stripping, you can image in each cycle 3 fluorophores, 3 antigens, not 3 fluorophores and 3 antigens. So on the same piece of tissue, you can look for probably around 80 antigens at the moment. All you need is a good Codex antibody panel, so this barcode technology is what you need to create the panel, or you can purchase it from the Codex website as well. They have many panels available for the tissue. And here is an example of the image. So it's multicolor image, many different antigens can be labeled in the same tissue. Although this is a wide field fluorescence imaging, it's not confocal, but it's still very useful. The disadvantage is, I would say, that it only looks at one very thin section. So, it doesn't give you a nice representation of the entire structure unless you process so many single sections. So the Codex works with, can be analyzed. Codex images can be analyzed with Halo software, which is equipped with high-plex module. And if you have questions about it, you can ask Connor. So we have many equipment available in our facility. We have 3 confocal 2-photon microscopes. We have Nikon spinning this microscope, which we use for live cells imaging. We have Keyence for wide field acquisition. We have 2 FLIM systems, confocal and 2-photon, which is integrated with the Leica. And also we have 2 Akoya phenocycler fusion. We also have lots of software available for you. IMARIS, it's very useful. It's actually very versatile software to deal with 3D, 4D images, and it's very intuitive to use. And we have Metamorph. You can also have Box for 3D rendering, Halo, IMART if you want to eliminate motion artifacts coming from live intravital imaging. So then, if you have any questions, please feel free to contact us. Dr. Dan is the director of the center, and Connor Goldbronson is my colleague who provides many services in the facility, but I would say that he's an expert from Codex imaging. So if you have questions, feel free to email us, or you can email our facility website as well. And we have our website, which I listed here, where you can find all the information about us. You can also find information about equipment, which we offer. Okay, and with that, I would like to thank you. And if you have any questions, I would be happy to hear them now.
All right, thank you, Gosia. I always enjoy looking at all of the beautiful images that you provide from your facility. Does anyone have any questions? You can either put them in the chat or unmute yourselves and ask. Maybe just a logistic question while people are thinking of their own. So if people want to get started, do they just contact you first? Do they go to iLab, or what's the way if they want to?
Yes.
Project?
The best way is to contact us, and then we can set up a meeting. And sometimes we just ask for details about your experiment via email, and then we set up a meeting. So then we give you instructions how to register in iLab, which is our reservation software. And if you need training, we set it up based on information which you provide, because it's very difficult to choose a correct microscope without knowing what type of imaging you're interested in performing.
Anyone else have a question? I think you went through things very thoroughly, so it appears not.
All right.
So thank you so much for presenting, and thank you everyone for coming.
Okay, thank you so much.